Okay, so, all right, cool. So today I'm going to be going over the next steps after the blur where you have your table and you're ready to start going through the core metrics and everything else below that you can see on the Qiime tutorial. So, yeah, let me just showing you guys here. So we finished the blur, right? It spits out the table, it spits out the rep seeks. I merge everything, and the tree you don't have to worry about because we have the merge tree, and that will work for your subsetted tables. So when you scroll down the tutorial right here where it says alpha and beta diversity analysis, this is the first step where you're going to start. And you should have all the files that you need to start this step. So this step is basically spitting out a bunch of different metric files that are used for alpha and beta diversity analysis. I highly recommend reading up on what these terms mean, alpha and beta diversity analysis. That's super important for just understanding kind of how microbiome data is analyzed from the sort of first go through. And yeah, they kind of explain it here, but I highly recommend just Googling it, and you'll, like, Google should give you a couple of hits that will be useful to read. So once you've done that, this is going to be the first chunk of code that we're running. So first, I'm going to go to Visual Studio Code, okay, whatever you guys are using, right? You should have the 4 files in one folder so it's easy to use. So here I'm in Final Files. I type in ls, and we can see that I have the metadata. That's what everyone's using, right? This is the merge table. This is not what you're going to use because you have your subsetted table. So here I have the subsetted mammalian table as an example. And then I have the taxonomy file. Again, we can all use the same file, so don't worry if that it's not saying bird taxonomy or fish taxonomy. We can all use the merged one. And then same for the tree. We can all use the merged tree file. So with all of that, we should be able to run core metrics. So let's actually see so I mentioned GitHub Copilot, which has been working great for me. Let's see if GitHub Copilot can fill in the code. But if you're not using it, don't worry. You'll be able to copy this code. Okay, well first I have to activate Qiime. So obviously conda activate Qiime2 23.2. Bam, bam. For some reason mine does this weird thing, but it's eventually going to recognize it. And then, so Qiime's activated, and then core metrics should be spelled like this. And let me just double check. So actually Qiime diversity core metrics. So I spelled it wrong. So Qiime diversity core metrics. So just a quick little preview of how cool GitHub Copilot is. It's already recognizing the function I need. Unfortunately, it's not recognizing the files in my folder, but it will slowly learn that and actually use the right files. So I can just hit tab to complete this. We want to change the tree to be our tree, right? The table is going to be your table. So in this case, I'm using mammals, but you use your table. For depth, we're going to let's go with 1,000. I didn't really go over this, but for now, to keep it simple and see the preliminary figures, it's fine if everyone goes for 1,000. And for the metadata file, again, plugging that in, you can use the merge one for all the samples. It's still going to work on your subsetted data set because all of your samples will be in the metadata, and that's all that Qiime cares about. So here I'm just going to call this mammals at the end, just so we know. Sure, we can call it mammals too. And let me quickly just go down to an S run where I can activate, so I can get some gigs going, so I don't run this on the login node. Okay, I'm gonna have to quickly move this. Okay. So short, that should be fine. Maybe not. So conda deactivate. So I might have to deactivate to get memory. You probably won't be dealing with this if you're on a your local computer, so you can skip through this part. Sorry about that. Okay, so I think I have the job running. Cool. So now I can conda activate qiime2 2023.2. And I'm in final files, just double checking ls, all my files are in there. Scroll back down to what I wrote. So theoretically this code should run. Let's see if we get lucky and it runs correctly. Bam. So just while we're waiting for this, the first thing we're going to look at is the unweighted unifrac plot. So this is going to be the pcoa plot. And this might take a while, so maybe I have some time to just quickly give you an example of what it looks like. So unweighted unifrac basically means that, so unweighted means they're not taking into account the relative abundance of the microbes. So basically, if microbe A is there, that's good enough. If it's not there, it's recorded as not there. So it doesn't matter if 20,000 reads of microbe A are there versus 10,000. It just ignores that. It's sort of a yes-no mechanism. Whereas weighted unifrac is a way of actually counting how much of a microbe is there. But for some reason, unweighted usually works better. And so we tend to go with unweighted first, just to have a first look at our data. And so that's not what we wanted. So it's not letting us look at it, but yeah, it's going to take a while. So yeah, you get some QZA files, which you don't want. You want what you will want is the QZV files. So here's the unweighted emperor plot QZV. So this you can view in the tutorial just to see an example. And so yeah, it's a basic pcoa plot. This is what you're going to see with your data. And you can cut the metadata is very important because now you're going to see all the metadata columns that we could look at. So for our case, it's going to be like diet, class, order, genus, all that kind of stuff. This example for the qiime tutorial is human body sites, and so you can see how they separate really nicely based off of body site. So what you'll want to do is you'll want to look at as many factors as possible. Probably the first one I would look at is order and family, and see how that clusters, and then diet. So those are the three big columns that you want to be interested in, because that's likely going to show some separation, like herbivores, carnivores, or like felids and canines. If you're looking at mammals, like you'd expect them to cluster together. So those are the first kind of figures that you'll want to do. Every time you make a new figure with new colors, take a screenshot of it, because that's I think the first step is you're going to just screenshot a lot of these figures and send them to me in some kind of document, and we can make a decision off of that. So let's see if it's finished. Okay, it finished. Cool. So we have a core metrics. We can see all the files spit out. And so let's just wait on downloading the unweighted unifrac pcoa plot. And let's move on to the next step, just so we can get you sort of getting all the files out first, and then we can look at them. So going back to the tutorial. So the next step, so the core metrics gives us a bunch of cool stuff. One of the things it gives us is a faith-pd-vector-qza. This is basically a species richness file. There's two different metrics people use. One of them is the Shannon vector. That's a way to count species inside of a community. A lot of ecologists do this for, like, measuring how species rich the Amazon is, right? And so what microbiologists have done is they've taken all of that and applied that to microbiology to look at species richness of gut microbiomes, for example. Like, how many species of bacteria are inside of your gut? That's kind of actually a complex thing to calculate. And so Shannon's is one way, and then faith-pd is in my opinion a better way, because it takes into account the phylogenetic relationships between two microbes. So if you have a bunch of microbes in a gut and all of them happen to be close cousins, that's not necessarily as species rich as, say, a bunch of distantly related cousins, right? So, distantly related cousins all in a gut are going to be considered higher up on the alpha diversity or the faith-pd vector metric. So this will make sense when you see a plot. Actually, I'll just quickly show faith-pd-group-significance, right here. So you're going to run this in Qiime, and then it's going to give you a bar plot. In this case, it's body sites, so gut, left palm, right palm, and tongue. And we can see right off the bat, it seems like left palm and right palm are the most diverse body sites based off of faith-pd. So these have the most different types of microbes residing on the sites here, which is surprising because you'd expect gut to be higher. So that's kind of interesting. Cool. So let's just quickly look back at the code to run this. So we're basically calling on the faith-pd vector that got made, and we're using the metadata, and then we're outputting a faith-pd-group-significance file, which is going to show us the bar plots. So let's see if Copilot can actually predict what I'm going to write. So Qiime diversity alpha group, let's see if Copilot can actually predict this. Bam, bam. So it didn't predict it right away. Qiime diversity alpha group. Okay, that's good. Let's see the next line. Okay, nice. Look, so it's already learning. So now it knows the next line. It knows that I saved it as mammals 2, and then faith-pd vectors should be inside of that folder that we made. So that's pretty cool. Obviously, a lot of you probably don't have Copilot right now. Download it if you want. I left a link on how to do it. If not, it's not a big deal. You literally are just, editing this from the tutorial. So yeah, edit that, edit this. This looks right. You're using the same metadata file. It should be there. It should be outside of the core metrics folder. So no need for putting the folder name in there. And then visualizing it, we can put it back into the mammals 2 folder, and it's called the same exact thing except QZV at the end. So this should work. that might take a couple seconds to run. Okay, that was quick. Cool. So now you're either going to just have this in your local computer, or if you're using Quest, go to CyberDuck, and then we want to find these files. So we got our core-metrics-phylogenetic folder here. We want to look for QZV, so faith-pd. Make sure it's a QZV because that's the one you want. Quickly download that, and then we want the unweighted unifrac emperor QZV. Make sure, so there's a lot of like unweighted, some of them are distance matrices. You don't want that because that's what it used to build the emperor plot. You want the emperor QZV, so just make sure it's that one. Did I download that? I don't remember. Cool. Okay, so downloaded the faith and the unweighted. Awesome. So the faith will look exactly like that other taxa bar plot I showed. I'll pull up the, let's look at the unifrac real quick. So qiime, drag and drop. So we're using qiime view, drag and drop. Okay, cool. So interesting pattern already. It seems like there's 3 groups. Let's see if it's because of the primers. Yeah, so it seems like we do have this primer issue that me and Diego are working on. So hopefully this will be fixed and we'll have a new metadata file that can clean this up. But let's see if we can look at class. So obviously it's all mammals. Let's look at order. So nice. It looks like we have some cool separation with order. Except the fact that primates, let's see if, yeah, okay, so primates are completely separating. It seems to be because of a primer, like being V3, V4 versus V4. So this is an issue that we're gonna have to work on later. But for the most part, yeah, this looks pretty good. Blue is carnivores. So it looks like the carnivores are mostly clustering here. The browns are rodents, which seem to be clustering together. And then we have artiodactyls as well. Cool. And then if we go over to, so I think first step, go to order, have this, and then just to make it look nice for the screenshot that you're going to send, I would go to axes first, switch the top one to black, and then the bottom one to white. And this looks just way better. We don't want the third axis just for like simplicity. So you can actually hide the third axis and make this 2D. And then you can first go to save image and save it as a PNG. And this will just give you a really nice high quality, because you can see it's kind of fuzzy and weird here. That will be high quality, that figure. And then just you can attach, you can put it in PowerPoint or whatever software you want and attach the labels. This is for like sending when we want like something that looks semi-nice to have an overview of your data. And then download the labels. The labels will just give you high quality labels that you can append to, yeah, like for me it opens up in Illustrator and you can add these right next to your PNG file. Cool. So that's how you get the unweighted. And then let's just quickly look at the faith-pd. So this is by order, which is already what we kind of were interested in anyways. And you can just see like it looks like perissodactyls, these are, a lot of them are herbivores, have the most microbes in their gut, you know, on average. Also Proboscidae, which I'm not even sure what that is, but primates are also up there. So this kind of makes sense because carnivores should be low, herbivores should be higher. So, and artiodactyls over here, these guys are also herbivores. So on average, herbivores have more microbes in their gut than carnivores because carnivores have really short guts that just process meat very quickly. So this makes sense. This is cool. You'll probably just take a screenshot of this, save this on a Microsoft Word document, and you can download an SFG file as well to hold like the vector. It'll be a little more high quality. But what you also want to do for sure is download the raw data as a TSV, because for papers, we don't really want to publish this in our when you start writing up your paper. You're gonna want a nicer figure that we edit in R, because or Python, because it will be much higher quality. So the TSV will be super important to have. This basically just outputs all the actual numbers per sample. So just save that as well. And then let's see, what else can we do? So we finished the alpha group. I believe the next step would be beta. So they have evenness. Don't really worry about evenness. It's something we could explore later if we want. Faith-pd for now is fine. The beta group, I would honestly hold off on the beta group for now. If you did it already, great, keep the files, because we will need the beta group. But the beta group is basically a way of testing the significance of clustering per some kind of column variable. So in this case, it was body site. Our case could be diet, But the thing with this is it makes sense for you to kind of talk to me, and we can pick some columns that make sense, and then you can start doing these tests, or else you're just going to do them on a bunch of them, and we might waste some time there. So again, here you can pick different distance matrices to do this. We're probably going to stick with unweighted distance matrix throughout the whole tutorial. Alpha-rarefaction. This is actually important. So honestly, we should just run this. Let's see if Copilot can actually predict. So qiime diversity alpha-rarefaction. All right. So qiime diversity alpha-rarefaction. Was that how it was? No, it was with the dash. Let's see if it predicts. Cool. So it does it on the right table. Does it give me the metadata? Yeah, wait, do we need the tree? Yeah, we need the tree. Max depth, we want, crap, I forgot. Yeah, so we want probably 4,000, I would say. We can go 6,000. Metadata-file, got it right. And then why isn't it doing, okay, core metrics, alpha-rarefaction, QZV, cool. I like to sometimes add mammal, because once you do a lot of these, it's nice to have descriptors, because then you're going to get, like when you download it, it's going to be like the same file over and over. So yeah, cool. So that should run. So this file, the alpha-rarefaction file, is important because this kind of shows you what happens at different sequencing depths. So to remind everyone, for the core metrics, because every sample has its own number of reads. Some samples, maybe they got lucky in the sequencer and they have 100,000 reads. That's great. right? 100,000 DNA sequences came back for sample A from a mouse. And then say for a snake, maybe, it's the digesta was a different material, maybe mucousy. It didn't run that well through the sequencer. You only get 150 sequences come back. So basically what you end up having is a bunch of samples, and there's a huge variation in how many sequences each sample has. And that's a problem when you're doing stats, right? So what you do to compensate for that is when you run stats, you actually slice at a sequencing depth and you normalize across the samples. So here in the tutorial, they do 1103. And so all samples are getting analyzed at 1,100 sequences per sample. And so if it has more than that, it gets thrown out, basically. So this is just a way to normalize the data. And so this alpha-rarefaction curve is a good way to help you with that number. So this is the tutorial data, and we can see at 1,000 we have a certain diversity, right? And it's actually kind of problematic because if we go up to 2,000 reads, we see that there's a bunch of diversity that's not being captured, right? Ideally, you want things to level out where you had determined your cutoff, which was 1,000, right? So let's see where ours cut off. This is also here. So the reason they picked 1,000 is because you lose samples. So that's the downside. So yeah, there is diversity you're losing by not going more than 1,000. But here are the number of samples as you increase sequencing depth, and you can see that there's a crap ton that get lost because they didn't have over 1,000 reads to begin with. So you have to counteract the okay, I want to capture more diversity and analyze it, but also I don't want to lose samples. So okay, so it's still running. That's going to take a while. Let's just go over the next step while it's waiting. So this is a curve. It's just nice to have this curve to kind of confirm the sampling depth you chose, and maybe if you see something different here, you can sort of modify that. This honestly should be before the core metrics, in my opinion. And the next step is going to be the classification step, which you're lucky because this I already ran and it's actually going to work for your data, which is awesome. So you don't have to run this. This is very computationally expensive, so that's good. So you can use the taxonomy that I made. So just know that this step is here. You basically use the rep seeks. These are the stuff that got spit out from deblur, right? Deblur gives you the table, but also the rep seeks, which are all the sequences. And then you use those sequences to map them to a database. And this thing's called a classifier because that's basically what it does. And it actually uses machine learning in a way to learn and classify your sequences for you as actual microbial taxa. So, Staphylococcus, for example, will get mapped to a specific sequence out of the thousands and thousands that you have in your file. And so they all get identified. And they all get merged into taxonomy QZA, and then you can view them in taxonomy QZV. So here, this is the feature ID. You might have seen this. This is like the codes that you'll see in your table. That's basically all the sequences just get this kind of code name. And then here are all the taxonomy that gets mapped to each feature ID. So this one happens to be a mitochondria, which you don't want in your data because that's a little problematic. This one is Akkermansia mucinifila. That's cool. This is like considered the next gen probiotic thing, that it's like correlated with Olympians and very healthy people. And then you'll see that sometimes a lot of features don't get identified to species level. You're going to see stuff stop at genus. and it just couldn't get mapped to the species level on the database. But genus, honestly, for a lot of analyses is good enough. This one only up to family. So there's going to be huge variation in how well it got identified. And then there's a confidence level as well, as like how sure was it that it's actually that species. So anytime you want to explore a certain species, you can use this table to see if it's there, because it actually has if I move this, yeah, it has a search function. So I can search Mucinifila, for example, and it will come up. So that's really cool. All right, so it should be done by now. Cool, it's done. Let's quickly download it just to see, because I'm actually curious. I haven't done this before. So am I missing something? Where is it? Alpha. Am I verified? Verified table. Oh, sorry. Okay. There it is. Cool. And then we want to go back to qiime2 view, plug it in. Already we can see that it's not ideal. We can see it shooting way up past 1,000. Let's select by order. So yeah, here's 1,000. This is where we cut off, right? So if this is the case where we see more than like 40, so at 1,000 we can see it's this one, the red one, which is perissodactyl, hovers around 50. And then if we went up to say 4,000, it goes to about 70 or 75. So that's actually not too bad, What I think we'll do is, if we do see this significant increase, because it's only for a couple of them, and then a lot of the other ones level out even at 500, they're leveled out. So it's only like the herbivores. I'm guessing these are the herbivores, because herbivores usually have tons of microbes. If that's the case, for now, we'll just stick at 1,000. And if for some reason we're having trouble analyzing data, we can go back and do another one where it's 3,000. But the thing is, here we're going to lose samples, right? So here are the number of samples. Here's 1,000. And then if we go to 3,000, you can see we start to lose some samples. It actually is it's not that bad for mammals, at least. So we can actually maybe even go up to 5,000 for mammals. Yeah, that's something to consider. So yeah, let me rephrase. If you do see, so at 1,000, if you still see a bunch of diversity going up, and then you come down here and you don't see that you're losing that many samples as you go past 1,000, I hope this makes sense. This is number of samples. If it looks like this, kind of flat, and not like that other graph we showed where they spiral down, then you can totally go back and increase your core metrics to, here I might even go like, I guess, so there's this green line that goes from like 10 to 0. So that's problematic. So I guess we're going to go right here before everything looks to stay the same, and we only lose a couple of this light blue line. So I would go up to 2,500. So this is something you can obviously screenshot me and we can talk about as well. So yeah, this is how we use this plot. if you've stayed this far in the video, then a little tip, I guess, would be to run the alpha-rarefaction step before you run the core metrics. Or if you had already been following along, then just, yeah, rerun it if you feel like you need to. So cool, we're done with that step. Now go back to the tutorial. And so because the taxonomy is finished, then really only the next and final step before you and I have a meeting and discuss like how best to move forward, because differential abundance is sort of, we need to figure out what exactly we want to analyze across the samples. Taxa bar plot is another important figure. So that's what we're going to want. Let's see how good Copilot is again. So qiime taxa. Let's see. Bar plot. Cool. Yep, that's the right table. That's not the right taxonomy. So bummer on that. That's another project I was working on. So taxonomy, and then we're going to want the GMTOL taxonomy too. There we go. We're going to want that metadata file, and then we're going to output it as a text. That looks great. Cool. So let's see if this works. So we'll just show them, while we wait, we'll just show this sample one. So they have about like 15 samples or 20. Yeah, so taxa bar plot is great. You can view it in qiime. It uses your metadata to allow you to sort the samples in different ways. Obviously, the easy thing here is we can change your rank. So this is by phyla, and you can see all your samples on the bottom, and then we can go down to like, family, I believe this is, just to go more in diversity. You can already see it's probably a good guess that these are different than these, right? And so what's cool is you can actually sort the samples by a metadata column. So let's do body site. In your case, it might be diet, could be interesting, or order. And so it will actually label all the samples, which is really useful because before this you had to like take it into Excel and relabel it yourself, and it was annoying. So yeah, we can see all the gut samples are clearly different from the skin samples and the tongue samples. So that's really cool. So this is what you're going to want to do. There is a problem though, because this is 20 samples. If you're working with fish or reptiles or even mammals, I think we might have like hundreds of samples, right? And so they're not going to fit. So let's actually see if that is what is about to happen. So let's just it has to be done. So let's download taxa bar plots. I mean, might not be done. Yeah, it's done. Taxa bar mammals too. So right there, get that downloaded. I'm guessing it's going to be very messy because we must have had at least 100 mammals, right? So it's not finishing or something. Wow, 50 megabytes. Yeah, this is going to be huge. So let's see if we can even view this. Yeah, so okay, so it finally loaded, but you can see that it cuts off, right? So this is not ideal. So if you already had made this plot, that's great. Keep it, it's fine. If it looks like this, then go back, and here's a trick that we can actually do. So instead of looking at all the samples, you can actually merge samples together. So it's a function called feature table, I believe it's group. Yeah, group. And so we can group the mammal table. You want to do it by sample. You can merge by features too, which is something you probably won't end up using. We're using the metadata, and so the best column to merge samples by is probably going to be order, I'm going to imagine, because class is mammalia, right? In this case, I believe birds is already class as well, so that you can use order. Reptilia, you can use order. And fish, you can use order as well. So yeah, order should work for all of your tables. If it doesn't, you may pick class or go down to family. It just depends. You might have to make a couple of these before you figure out the right one. And then we're going to do sum. So sum means it's merging the reads, not taking the average, but actually just adding them up all together. Hitting tab. And then grouped, the output is group table. And so it's literally just a new table, but saying that it's grouped. So now, instead of having hundreds of samples, you're going to have maybe 20 samples. So like, carnivora is an order of mammals, and so that's going to be one bar in this taxa bar plot. So all the carnivore samples are merged into carnivora. Artiodactyla is going to be another bar. You'll see when it actually runs. So group table. And it's not going to work because I forgot one of these. Always remember your slashes. Okay, so this should work fairly quickly. So this is going to merge everything together. And then while we're waiting, we can actually just start writing the taxa bar plot code while that's running. So this is the new taxa bar plot. We're going to be using this new table. Because this is the merge table, right? We're going to use a taxonomy. We're not going to use the metadata. And I'll explain this later. Basically, your sample IDs become the order names because we merge by order. So you have to make a new metadata. But what's really nice is Qiime allows you to make it without the metadata, which is actually, you would think this is trivial, but this is a great feature because it sucks to make a new metadata every time you group. So let's call this taxa bar prop plots mammals grouped QZV, and then run that. And this should work. And then while that's running, let's just double check the tutorial to see if we are missing anything. So there is a differential abundance component. So this is the end of the tutorial. So I just want you guys to see like how easy this stuff is. We basically almost did the entire tutorial in the last 30 minutes. So yeah, this is so incredibly easy, which is why some of you were saying that you like want to try to get this done by the summer. You literally could do this on a Saturday, or during the week if you're trying to keep your summer Saturdays open. Yeah, so we can have figures within, by the end of this video, you should already have the figures that you're going to need for us to have our next meeting and discuss what the next plan forward is for the paper. So yeah, this is I'm pretty excited because we're gonna get some very fast data. Okay, enough chatting. So Taxa bar plots is made. Quickly download it again. Taxa bar plot, where are you? Did we make it in the, okay, grouped, right? There we are. This is going to be actually a big file. Oh, it's only 5 megabytes this time. Interesting. So it's actually a much smaller file. And then we go over to qiime2 view. This is grouped. Cool. So now look how simplified this is. So now we have like 8 or 9 or something, right? This is so much nicer to see. Let's look at the phyla and let's see if it makes sense. Yeah, so they seem to be pretty different from one another. The herbivores should be a little more diverse than the carnivores. So carnivores are already looking about like, not as diverse, right? And then perissodactyl and artiodactyl, these are like elk and stuff. They look a little more diverse. It's hard to say just by looking at this. But they should have more bacteroidetes, proteobacteria. So yeah, the purple one should be higher in the omnivores and carnivores. So rodents and primates are omnivores. So yeah, this does make a lot of sense. We have bacteroidetes and then we have firmicutes, should be high in all of them. It's interesting that it's low in bats. So overall, it makes sense. Mammals should be similar, but then there should be the differences between certain taxa. Cetaceans are carnivores, and they look very much like these carnivores. They have a lot of fusobacteria, so it's starting to make sense, and that's good. So this is a really nice plot. This is almost publishable. I think what I usually do is I download the CSV file and put this into R. This will be like phase 2 of sort of figures and publishing and writing up the paper. But for now, you could just download the SVG and it gives you a high quality picture. And then the legend is a separate one as well. So yeah. And then I even forgot so because we didn't upload with the metadata, you can't sort them by diet or anything like that. So if you do want to make a new metadata, it's going to have to be literally you just copy. Let's just quickly show you that step. So it would literally look like this. Sample ID, and then say you're interested in diet. You would literally just start copying the names. So I'm not going to copy that long one, but Chiroptera, Cetacea, Carnivora. So you have to spell it exactly how it is. Carnivora is obviously a carnivore. Artiodactyla is an herbivore. So for this, you'd have to go back to the old metadata, our original one, to look at what the actual diets of these groups are. And yeah, basically make that new metadata column, re-upload the taxa bar plot with this, and then you would be able to sort by diet. So then you can have a taxa bar plot that will show you the herbivores first, omnivores, and the carnivores. I think most of you should be able to do that without me having to go through that. If not, quickly just message me. I'm happy to go through with you. But yeah, I mean, basically after this, you have 90% of the figures that you need for a publishable paper in a reputable journal. Yeah, this is pretty amazing stuff. So like the one last figure might be a differential abundance analysis that can be sort of the icing on the cake. And that's something that I can help you with in the fall. So I hope this was helpful, and sorry if it was kind of long and felt like, I was working through it with you, because I was doing this for the first time and it's been a while. But yeah, I'll see you guys. Have a great summer, and I'll see you guys in the fall. Bye.